Can you hear me? Is it okay? Do I speak loud enough? Yeah. Okay, thank you. So thanks all for coming, and I really want to thank the scientific committee for inviting me, because this is so nice. It's my first international conference alive, and I really enjoy having all these side interactions to the talks. So today I am going to focus on in situ NMR studies that we've been running for energy storage. And I just want to start with a quick reminder of the two main devices, the two kind of devices that you can have for electrochemical storage. The first one you know very well are lithium-ion batteries. They are based on redox reactions, and they can store relatively high energy, so they have high capacity, but one of the issues are the relatively slow charges. And this is an issue, especially for cars. You don't want to wait 5 hours at the station. The other devices that you may not know about are supercapacitors. So they're electrochemical double layer capacitors, and they're based on charge organization. So you can store the energy much faster, but of course, the capacity that you can store is smaller. And as we're all aware, with the energy crisis and climate crisis, we need to use more and more renewables, and we need to store them because of their intermittent nature. And I don't need to convince you that NMR is one of the techniques for characterization that is essential. So our goal is to use a combination of spectroscopy and imaging to characterize these kind of devices and to look at them as a whole. So to follow really what is happening inside the battery. And so they're actually quite complex. So let me get the mouse there. So you have different parts. It's a very heterogeneous device, but they're all based on the same principle. You have a positive electrode, a negative electrode, some electrolyte, usually liquid, that is soaking everything. And the ions can equilibrate the charge by passing through the separator, and the electrons go through the outside circuit, and that's your current. So in the case of lithium-ion batteries, there are redox reactions at the positive and at the negative electrode, and at the same time, lithium ions are moving back and forth. So we have lithium everywhere in there. And if you look at an NMR spectrum of lithium, what you get is actually a superposition of all the two contributions. It's actually very difficult to tell anything out of it just by looking at the spectrum. For supercapacitor, it's a bit different. The two electrodes are the same, it's just their polarization that is different, and it's just the organization of the ions. And so again, you get lots of superposing peaks and also some shifts that come from magnetic susceptibility changes. So for running, what we aimed at was to use some imaging capabilities of NMR to disentangle these spectra and be able to tell things about the positive and the negative electrode, or the ions in the positive and the negative electrode. So the setup we have that we designed at our lab was with this kind of electrochemical cell. So it's the same principle as a coin cell, except that we're inside a plastic casing, and we have a sandwich of electrode, the positive electrode and the negative electrode. And we designed this back in 2014 when there were not so many people doing in situ NMR. And we designed it so that it could fit inside a saddle coil of a diffusion probe from Bruker, so that we would have a strong pulse field gradient along the Z direction, and that we could localize the spectra. And so this did fit inside a diffusion probe, and then you bring it into the magnet. And so the battery is located here at the center of the RF coil. You connect it to a galvanostat for charge and discharge, and at the same time, you record the NMR spectrum. So of course, we're not the first to do this. This has been developed by Le Tellier and expanded really by Claire Gray. And so just to echo on Joanna's initial presentation, this is what it really looks like inside the lab. So this is the DIF50 probe. We remove the gradient sheath so that you can see what is there. And so the electrochemical cell is in there. And we connect it here at the bottom for one electrode and at the top for the other electrode. And then we remove the dew wall that is inside that is usually used to control temperature, and we replaced it with the electrochemical cable so that it goes through the probe, and it's actually very well shielded, and we don't get much problems of noise by doing it so. And so then you put the probe inside the magnet, you connect it to the galvanostat, and you can charge and discharge the battery as you measure it. So I won't spend a lot of time on this, but this is one examples where we used a combination of spectroscopy and imaging to get information on positive electrodes. So it was an oxide, lithium titanate oxide and lithium cobalt oxide. These are the two electrodes. And if you do chemical shift imaging, so this is the Z position, this is the spectrum. If you run standard echo-based chemical shift imaging, then the only thing we can see is the electrolytes inside the battery. So we worked on using these strong pulse speed gradients, and also we went back to smart techniques that were developed actually by Ordage in the 80s for very short C2 solids to be able to retrieve the signal from the positive and the negative electrode, and then we could apply it to these thick electrodes following what was going on as we were charging and discharging it. And by following the width of the spectra, we could follow the redox state of the electrodes and identify some limitations, some kinetic limitations inside the batteries. And with Charles-Emmanuel Dutoit, we've been working on going faster to get some resolution and to follow what is happening, some kind of relaxations in these electrodes when they're cycled too fast. And so that's something that is actually very useful for identifying the limitations in the thick electrodes. So what I want to spend a little bit more time today is an application we did on supercapacitors, and this is mainly work by Renima Oukali, who was a PhD student in our group. So as I said before, the supercapacitors are based on an electrical double layer forming at the surface of the electrodes as we charge them positively and negatively. So here we have nanoporous carbons as electrodes, and this charge organization stores the energy in the form of a capacitance. Using the capacitance, and this capacitance is related to the distance between the ions and the surface. So this looks quite simple, but actually the electrodes here are not plain. They're actually much more complex. There are like different types of pores and different structures of carbons in there. And you can see that this is the capacitance as a function of the average pore size. And there was something really interesting going on with tiny, tiny pores with a large increase in capacitance. And this was related to the removal of the solvation shell for the ions to go in so that they would get closer to the carbon and give a higher capacity. So of course, this is a very interesting thing for NMR because the ions that we're using have NMR active nuclei, and we wanted to know more about what the ions do inside the supercaps. So there had been ex situ studies. So ex situ studies, you would charge and discharge a supercapacitor, bring it inside the ex situ and inside the glove box, scratch it, measure it by magic angle spinning NMR. And so what could be shown is that for a negative electrode, so that's a pore, you have several ways to compensate for the charge. You can either have cations going in or anions going out, or both at the same time. And so ex situ, what was identified was exchange, but there was something a bit weird at the negative electrode that the acetonitrile would also move in the process. And so there was the question whether this was ex situ NMR because the acetonitrile is can evaporate so easily, or whether it was real. There had been another study 2 years later by John Griffin, Alex Force in Claire Gray's group, where they identified a slightly different mechanism with a bigger proportion of counter-ion absorption. But again, that was not the same carbon. It was a phosphonium instead of an ammonium, and they changed the geometry. So in that case, because they couldn't look at the whole device because everything would be overlapping, what they did is that they slide apart the two electrodes so that only one was inside the coil and was measured. So that was a like physical separation of the two electrodes. And so we decided to go with our electrodes to see what was going on. So the initial idea was to use this imaging capability, localization capability to follow the positive and the negative electrode in a regular design. But what happens again here, because you're doing these echoes, by the time you ramp up and you ramp down the gradients, the signal of the strongly absorbed ions disappear. And we do see shifted signals that correspond to relatively absorbed cations and or anions, but they actually don't change as we charge and discharge the battery. And this is because we're not quantitative. And the ISIS technique we used before for the batteries couldn't work here, mainly because the T1s are so long and we need to accumulate a lot of signal here. And so this would have taken too long. So what we decided to go was to go for pure imaging using ZTE imaging. So this time you run the gradient up, you give a short pulse to excite as broad as possible, and you acquire under the gradient. So that's frequency encoding. Now your field is proportional to the position along Z, and you can translate directly your spectrum, the frequency scale here into a distance, a position scale. So there is some blurring, and Michael Garr would explain that really well in his lecture. There is some blurring related to the chemical shifts, and in our case, this is about 70 micron blurring. But our electrodes are bigger than that. And what we can see is that we have profiles, so we can do this for protons to follow the cations, and with fluorine to follow the anions. And as at the beginning, when the supercapacitor is assembled and we didn't do anything to it, this is the kind of profile we have. So that's basically the concentration of protons inside this battery. It's distorted by all the interfaces and so on. But as we charge and discharge, we do see changes. And these changes can be followed both for protons and fluorine. And so to exploit and better understand these changes, what we did was to subtract the profiles at the beginning and when it was fully charged. And what we observe is that the cations are expelled from the positive electrode, and at the same time, the anions are absorbed inside the positive electrode. So this is confirming this exchange mechanism. And when we compare it for the negative electrode, what we see is that the contribution of the fluorine anions at the negative electrode is actually much smaller than at the positive electrode. And this is indicative that at this electrode, the mechanism is slightly different. So that was quite interesting. And then we confirmed this by discharging the supercap. And this time we do the difference in orange between the fully discharged supercap and the fully charged supercap. And you can see that this is quite reversible. And this is one of the things that is a characteristic of supercaps. We also checked what was happening with the solvent. So I forgot to tell you, but all of this was made with a deuterated solvent so that the protons from the solvent wouldn't disturb our subtractions. And we did this test also with protonated solvent so that to see whether we had a macroscopic flux or not. And as you can see, the profiles are much more intense because now we have contribution from the solvents, but the difference are of the same amplitude. And so this is indicative that there is no macroscopic solvent flux inside the electrodes. It does change inside. I mean, it doesn't mean that inside each electrode it's different. And so then what we tried to do was to compare different types of carbon. So this is a commercial carbon, and this is a carbide-derived carbon. So it's chlorinated at high temperature to expel the titanium chloride, and we end up with a nanoporous carbon that has very, very tiny pores. And by doing so, we could identify that the fluorine anions did contribute a lot more in these carbons. And so this gave information on the mechanisms. And so for the remaining time of my talk, I would like to show you what we've been doing for Ludivine Afonso's PhD thesis on the subject of low temperature and fast charge for lithium-ion batteries. So this is also something where we have really high kinetic issues. And so in this case, what is happening is that we get a capacity fade as we increase the charging rate or as we lower temperature. So what happens is that at the negative electrode, we get reduction of the carbon by an insertion of lithium. This is the normal reaction that is highly reversible. And now, if you go really fast or really low temperature, you get overpotentials that make this reaction possible. That's the reduction of lithium ions. And it does plate on the surface, and that's an issue because it can have different microstructures or react with the solvents and consume lithium. And this has been identified as the main source of capacity fade. So right now, how the constructors are avoiding this problem is by measuring in a battery, adding a third electrode to measure the potential of the negative electrode, and then they identify when the potential is below that of the lithium plus lithium zero couple, and they devise charging protocols that avoid this region. And of course, what they would like to know is whether there is actually plating or not, and whether they could actually go below and not have plating. Because this third electrode method is actually very dependent on how you put the third electrode in the battery. And so there was this question, can we do it by NMR? So this is what it looks like. What is the spectrum, the in situ spectrum looks like. So we have signal from lithium in the electrolyte and in the passivating layer. We have lithiated graphite here. And this time we don't need spatial localization because spectroscopically the metallic lithium is in a different place. And so we've been working on getting relevant electrochemical behavior. That's the most important if we're doing in situ NMR. And to be honest, I think we still have work to do on that subject. So what we did is that we used the cell we devised previously, because for plating, we know that compression is very important. Tests on pouch cells that are not compressed show that you get plating much earlier than what you would get in a real battery. And so we use commercial electrodes and protocols from our partners. So initially, so this is what you have to describe the electrochemistry. In blue is the voltage curve, charge, holding the voltage at 4.2 volts, and then discharge. In red, this is the current, and this tells you how fast you charge. So we always prepare our batteries in the same way, and we check that they do give what you expect. And then we had to change our setup for low temperature. So this time we cannot remove the dewar because we need to control temperature. And so what we did is that we went for a Bruker solenoid probe, which are like heavily robust to low temperature. And we removed the traditional RF coil, and we inserted instead a saddle coil that would allow us to have our battery, our electrochemical cell inside. And so the cold gas is coming from the bottom with a thermocouple here and everything to control temperature. And then we connect the cell inside. And this is what it looks like. We do have some small cables to connect it here. And then at the top, we have the electrochemical cable that comes out here. It's held here so that it doesn't get damaged when we move the probe. And here we have a cable that is connected to the galvanostat. And so this time we get a lot of noise from this setup, and this has been sorted. You need to ground your negative electrode. You need to protect. We put a Faraday cage around our unshielded connections, and we could reduce the noise so that you actually can see the lithium signal you're interested in. And so with this, we can start doing our time-resolved lithium spectroscopy. So we record one spectrum as we charge and discharge the battery, and we observe the metallic lithium region. And this is not discussed a lot, but actually here there are a lot of steps with approximations that you do. And so of course we had to phase the spectrum every time because we want to charge the battery quite fast in 15 minutes. So we don't have time to tune it every time. Even automatic tuning is too long. So the phasing, the tuning changes slightly and the phasing as well. So we had to rephase every spectrum independently. Then we had to apply a baseline correction because the positive electrode is very broad, we don't see it, but its redox state changes, and so its effect on the baseline does change, so we need to correct for that also. Then we worked on the integration and also on the uncertainty. We need to time label each spectrum so that we can interpolate it with the different timing grid of the electrochemical data. And then finally, we get the correlation with the NMR. And this is typically what you get. And so what we could see is that, for example, if we go for a faster charge, so this is a current here given in portion of capacity and time. And so what we see is that if we charge very fast, we get some metallic lithium showing up. And then that's really during the voltage, the constant voltage step that we get most of the lithium that is plated. And what was interesting here is that during an open circuit period, so you charge your battery, you disconnect it and you observe, then we saw that the lithium was actually not reacting. We could still detect it. And that's interesting because it can be retrieved as we discharge the battery. In green here, the amount of lithium is retrieved, not all of it, but most of it can be retrieved, and this can be seen by the relatively good capacity that is left. And so what we saw is that if we go faster, charge in 26 minutes or 30 minutes, then we get dead lithium. And so then I just want to show you what we did at minus 20 degrees. This time we go very fast because we have high overpotentials, and we had to hold the voltage for a very long time. And this means a lot of plating. Going back to 0 degree, we could retrieve most of it, but there was still a bit of metallic lithium left. So, I mean, my main message, and this is still work in progress, is that we want to do statistics. We really want to be able to be sure that what we observe on a single battery is meaningful. And this is what we've started doing here. We do see, for example, at 0 degree, that most in red is when we detect lithium. The lithium, there is a lot of dead lithium left as we discharge, while at 0 degree, we had a lot less. And this is really the goal of our work, trying to have these statistical, meaningful conditions for lithium plating. And so just to conclude, so we can use either pure 1D imaging or pure spectroscopy in some cases. And we really want to keep optimizing everything. So I want just to finish with thanking the people that have been working on this, and especially the NMR team at CEPTI in Orléans. And in case you wonder where we are, so we are in France, just 1 hour south of Paris. And we are part of this pan-European infrastructure. We have low fields with all kinds of interesting capacities, so please go and visit if you're interested. And I thank you for your attention.
Questions for Elodie, and thanks for your presentation.
Thank you, very nice talk. In your selected slides by 180 pulses followed by the imaging measurement, if I read it correctly, you apply a very strong gradient to do the slide selection. Does that correspond to give you a poor signal for individual slides? Could you comment?
Sorry, there is an echo. I cannot hear you very well.
The one of your beginning slides is where you show the you do the selected 180 pulses in the presence of gradient to look at the image slides in 1D imaging.
Yes.
So
you mean on the batteries?
Yeah, if I read it correctly, you're applying a very strong gradient pulse to do the selection. I'm just wondering about sensitivity.
You mean this one?
No, before that.
Yeah, the beginning. Okay.
Yeah, this one.
Yeah. Yes.
Yeah. So you're plotting a 24 Tesla per meter.
Yes.
So do you have a sensitivity issue?
So, I mean, yes, we need to accumulate. And so we need to accumulate, and this takes about 3 hours to record.
Okay, okay.
And we also have like quite thick slices also, because these are solid paramagnetic materials. So we actually only have 100 micron resolution. Yeah.
Okay.
So we still have quite a lot of spins in each slice.
Okay. It just have the number doesn't look quite. Anyway, do you have a poster or something that?
Sorry?
Do you have a poster somewhere or something?
No.
Okay.
But we can chat afterwards.
I'll chat later. Okay, thank you.
Any other question? In the interest of time, let's move on and let's thank Elodie for her presentation. Next, we have Wangshang Zhou from Louisiana State University.
Okay, thanks, Dr. Hayes. Good morning, everyone. My name is Wan Cheng. I'm from Dr. Wang's group, Louisiana State University. Today, I would like to introduce our new work on using solid-state NMR and the DMP technique to do the biopolymers interaction detection in plant cell walls. Nowadays, biomass materials is well known as a natural renewable resource for producing biofuels, or being the construction materials or chemical feedstocks. The plant cell walls are a major resource of the biomass. It includes the primary cell wall and the secondary cell wall. The complex carbohydrates, cellulose, hemicellulose, and pectin are the major component of primary cell wall. In the secondary cell wall, it also includes the complicated aromatic polymer, the lignin. So if we want to make better use of the biomass materials, we need to understand the plant cell wall structure very well first. In the traditional analytical method, we need to dissolve the biomass materials into the organic solvent or ionic liquids to do the further characterization. However, this process must perturb the physical state and the molecular interactions in the plant cell walls. So we want to avoid it. The solid-state NMR can solve this issue. We just need to pack the carbon-13 labeled plant stem into the rotor and collect a series of 2D and 3D experiments, and then we can get all the information that we want, just in native and intact cell walls. Our group has made several efforts on illustrating the primary cell wall information. Currently, the challenges is on the secondary cell wall. Could we detect the lignin to carbohydrate interactions, and could we get the lignin linkages information in secondary cell wall? Here, we employ these three kind of woody plants. Because the wood contains more lignin component, they all are uniformly carbon-13 labeled by this company. They just grow them by feeding them with carbon-13 dioxide day and night until harvest. Here, there's two hardwood, eucalyptus and poplar, and one softwood, spruce. So with the carbon-13 labeled sample, we can just start to collect NMR data. Firstly, we run the refocused J-inadequate experiment. From the spectrum, we can clearly see the two-fold xylene, the three-fold xylenes, and the cellulose information, include the interior one and the surface one. Otherwise, besides, we can also detect other carbohydrates information. From the inadequate spectrum, we can also observe the different hemicellulose composition across the wood species. For example, this one, the glucuronic acid only show up on the hardwood spectrum, and the mannan is a unique component in the softwood. Besides the CP inadequate, we also detect the DP inadequate and the RFDR experiment. All these three experiments can detect through one bond intramolecular interactions. They are very helpful for us to do the chemical shift assignment and to get the composition ratios in the secondary cell walls. Now, we can move our eyes to the lignin part. Lignin is chemically complex. So they are made up by these aromatic rings and connected by the lignin linkages. In our inadequate spectra, we can clearly get the aromatic rings information, and we can see in the hardwood, eucalyptus and poplar, the S unit are dominant, but in the softwood, it only contains the G unit. When compared to our previous work, we know the H and the FA unit are rich in grass, but we cannot see them in our wood plants. The lignin aromatic rings are connected by these different kinds of linkages, for example, beta-O4 and beta-bit. Here, we use RFDR experiment to successfully get the linkers informations, such as this one, the S1 to A alpha on the left top side, and also for the A beta to A gamma, and so on. So we can get this linkers information and its untreated plant samples in the real in situ environment. Another important thing is we can get the unprotonated carbons information in the lignin. Why it's important? In currently lignin research, the solution AMR, especially HSQC experiment, are a major method to detect the lignin information, but it's hard to give us the unprotonated carbons information. The solid state AMR can do it. Now we can try to find the lignin carbohydrate interactions. We use this Doppler-gated PDSD pulse sequence. We selectively detect unprotonated carbons information and suppress the protonated carbon signals in the carbohydrate. Then we got this beautiful spectrum, and we can see hundreds of the intermolecular interactions. For example, xylene to cellulose, and xylene to lignin, lignin to cellulose, and lignin to xylene. All these different interactions, we can get the information, see which carbon is talking to which carbon. Now we can just summarize the interactions into this figure, and we use the blue bold lines to represent the strong interactions, and the pink and the black one as the medium and the weak interactions. They give us the basic information about the interactions. The result shows in the wood samples, we have more abundant lignin to cellulose interactions when compared to grass or Arabidopsis. Now we can give a zoom out view of our PDSD spectrum. We can notice the softwood one looks specific. The short range 0.1 second mixing time spectra can match the long range spectra very well. That means there must be homogeneously molecular mixing in the softwood, but doesn't happen in the hardwood. To get more details about the interactions, we look for the DNP's help. The DNP is a very important method to get the sensitivity enhancement in plant cell wall study, especially for the natural abundance plants. So our colleague Fabian will give a poster presentation about it to talk natural abundance samples. Let's back to this project. Here we use the biradical amipole. The gyrotron gave the microwave to irradiate the amipole, and we can just transfer the polarization from the electron to the proton and then to the carbon in the plant cell walls. Here, we got a 24-fold sensitivity enhancement. With this big enhancement, we can collect the DNP-assisted aromatic additive spectra. In this spectra, only the signals of carbohydrate that rarely contact with lignin can show up. When compared to the normal PDSD, we can notice that this peak is missing. Its IC peak is the deeply embedded cellulose microfiber peaks. That means all the xylenes and cellulose can bend to the lignin except for the cellulose core. Besides, our collaborators also did a DFT calculation to talk about how the lignin and the carbohydrate interactions happen. For example, for the 3-fold xylene, it can form a packet to enclose the aromatics. Now we want to get the hydration level of the wood samples. We use this water-edited spectrum with a DAR mixing time and get this 1D build-up curve and this 2D water contact maps. Both of them show us the poplar is the worst hydrated wood samples. And when compared to the different biopolymers, we can see mannan is most hydrated one, but cellulose is the hydrophobic one, no matter in which kind of wood. Solid-state AMR can also tell us about the dynamic information in the wood samples. We can use the carbon P1 relaxation to get the nanosecond time scales motion. Here, the mannan is the mobile part, the cellulose is the rigid part. And in the microsecond time scale, we can use the proton T1 row experiment to get the information. The cellulose is still the rigid part. No matter the nanosecond or microsecond, the spruce, the soft wood, is always the faster relaxation part, wood samples. Now, there's still one question need to be answered. For simplifying our shaping process from Europe to the US, and our wood samples has a pre-freeze drying treatment and before shaping. After got it, we just do the rehydrated and collect the AMR data. So does this lyophilization rehydrating process affect the result? Then we order a new batch of spruce samples. It's a fresh one, never dried. We collect the major 1D and 2D experiment and get this data. We do the freeze drying overnight by ourselves and rehydrated them and collect all this experiment again with the same parameters. And then we can see the result matched well. That means the answer to this question is no, it doesn't affect so much. Now we can just wrap up all the AMR findings to give this illustration model to show all the major findings. In the hardwood and softwood, we can see the lignin to cellulose contact, and in the wood samples, there's more carbohydrate and lignin interactions, and there's a better molecular mixing when compared to the grass model. There is still two major factors when we talk about the future research in the plant cell walls. The first one is the sensitivity enhancement, especially if we want to use the natural abundance samples. So the answer should be a mass DMP. Another one is the resolution enhancement for the structural complexity of plant carbohydrate. So one solution is increase the dimension of the AMR. Currently, we just developed a new 3D CCC experiment. We just add the chord block after the inadequate. And we can got a double quantum to single quantum 3D spectrum. Here, it does increase the resolution of the carbohydrate. We can get a better interpretation of the cellulose polymorphic structure. If you are interested on this project, you can just visit our poster section number 9 and number 15 to get more details. In the end, I would like to thank my supervisor, Dr. Wang, all the Wang group members, and all the collaborators, and all the funding support. I would like to say special thanks to the ENC conference. This is my first ENC one, and it's also my first in-person talk in the conference due to the pandemic. And everything is amazing, it's impressive. I'm really enjoying here. So thanks for the great opportunity and the good organization. So thanks for everyone's attending, and I would like to accept any questions. Thank you.
And any questions for Mr. Zhao? And please, I like to encourage students and postdocs, especially in these sessions. Yes, Carl.
Thanks for a nice talk.
Thank
I was struck by the one figure you had that showed the interactions in the different materials and how different they were, which ones were the strongest ones. And I was just wondering if you had, if there's some rationalization of why those were different in the different materials.
You mean this one?
That one, yeah.
Okay, so just a basic idea, so is because in our previous work, we also did a grass investigation. It's totally different with wood one. So here we can just see more, lignin to carbohydrate interaction, especially lignin to the cellulose. And we also, each color represents each component, and we can see which the part of the materials can contact with other materials, just to give us, we can say, a statistic idea about it. Thank you.
Christian. Or Carl, did you have a follow-up?
I was just wondering if there's, if there's some correlation with properties of the materials, or some sort of understanding of the impact of those differences.
Yeah, so I'm not sure if I really catch your question, so maybe this figure can give some ideas about it. So, but the plant cell walls inside environment is really complex. So we can just through our AMR data or evidence to give some suggestion, but we do not have enough knowledge to understand why they have this kind of interactions or something.
Very nice talk. I missed one point. What is the DNP juice you've used in your DNP experiments?
The glycerol and the D2H2O 6:3:1 ratio.
Okay, and what by radical amipol or
Amipol.
amipol? Okay, did you try some other solvent?
Yes, we also tried, but not in this study. So currently we are also tried DMSO, and we tried we can say a matrix free, just the D2O, H2O, or only H2O. We are also studying in this field to try the best juice combination and the best radicals in the, we can say, cells. And currently we are trying a simple POC.
And looking at the spectra you obtain with the different juices, do you have differences in chemical shifts?
So actually, we also have a publication talking about it. We didn't see much, or we can just say no difference, and only the sensitivities.
Thank you.
Yes, thank you.
Hi, very nice talk. I had a question. So you would imagine in a biological material, it's not going to be the same throughout. So it's likely going to be heterogeneous. How do you account for that as you're mapping your interactions?
I'm sorry, I didn't catch the first part. Could you,
Yeah,
repeat?
so in biological materials, they're often fairly heterogeneous throughout. So you're going to have maybe different concentrations of lignin in one part of the material versus another. How do you account for that heterogeneity as you're mapping your interactions?
You mean for the model part?
Yeah.
Okay, so here, we can see from this project, we got almost hundreds MR connection data. And that's showing the water contact and showing the intermolecular interactions. And for example, which carbon to which carbon. And all the details to showing just to give us some imagination to see how they combine. And we can only find maybe this situation can match all the MR data result. Otherwise, it will just give some violation to our MR result. So we cannot say it's a real one, but it should be the best fit one for current data.
Right. Thank you.
Thank you.
With that, okay, with that, let's thank our presenter. And next is Anna Parker, who is coming to us from Envision.
All right, thank you, Sophia, very much for the introduction. So as she said, my name is Anna, and I'm working with this company called Envision Imaging Technologies. We're a startup company based in southern Germany. And I'm really excited to share with you some of our work. So just to give you an idea of the type of results that I'll present in this talk. So we've been working on a technique for producing hyperpolarized NMR spectra based on optically polarized crystals. And so this is just an example of what our technique can do. So basically we take an intermediate source of polarization, which is highly polarized proton spins on naphthalene molecules. And we can transfer polarization from these proton spins to protons on a target molecule to get a hyperpolarized solution state spectra of this molecule. So this is just one nice example for this molecule papargyl acetate, which we studied, where you can see we get enhancements of 200 to 2600 over 60 megahertz, or 20 to 130 over 400 megahertz. And so we tested this technique at a few different magnetic fields. So you'll see throughout the talk that I'll just present enhancements for a few different magnetic fields. But this is sort of the type of spectra that we can produce with our technique. So just to give you a brief outline before I get into the talk. So first I'll go into sort of our thinking, as well as how our technique actually works for producing hyperpolarized NMR spectra. Then I'll go a bit into the phenomenology of the technique. So interesting things that we learned while developing this and how we handled them. And then finally, I'll just conclude by introducing some of the things that we've been working on after our first demonstrations. Okay, so I'll just get right into it. So Envision has been interested in hyperpolarization in general. So hyperpolarization is this body of techniques where you can actually modify the population of your spin states to produce a higher population difference or a higher polarization. And for NMR, what this gives you is just a much larger magnetization to work with that you can measure with your NMR spectrometer. And so some of you may know, like even with state-of-the-art spectrometers, your polarization is typically on the order of 10 to the minus 5. So hyperpolarization offers possibly orders of magnitude gain. And so this can make the difference of getting better information or just getting information where it just didn't exist currently. So there are a lot of really nice demonstrations of hyperpolarization and also really nice commercial products that do this really well. So a lot of these are definitely not the only sources of polarization that are used, but stable radicals are often used in polarizers. So these are electrons that thermally polarized electrons that you build a high polarization by placing them in a large magnetic field and by cooling them to low temperatures. So typically in these techniques, you need a highly polarized source of polarization that you couple to your target nuclei and then use that to transfer polarization to your target nuclei. What Envision is interested in particular are these two sources that I've highlighted on the right. So we're really interested in what you can do with optically polarized sources. So optically polarized electron spin states. And two really nice examples of that are NV centers in diamond. So NVs are a system of trapped electrons in diamond, and pentacene doped molecular crystals. So pentacene is this highly conjugated molecule where you have a system of delocalized electrons that you can excite within the visible range. And you can also build these into single crystals and use that and basically fix the position of these molecules and orient them. So these are two really nice examples of electronic systems that you can optically excite and produce a highly polarized electron spin state. What's nice is that the polarization of this electron spin state is independent of magnetic field and temperature. So a lot of people are interested in these systems because possibly you can really reduce the hardware constraints that you typically have with thermally polarized electrons. So Envision really wanted to see what you can do with this. Okay, so just to give a bit more detail, On the right here, I'm showing an energy level, or like a simplified energy level structure of the pentacene doped naphthalene system. And just to give you an idea of how dynamic nuclear polarization works with triplet electronic states. And so what happens is if you pump this electronic state, you'll get some systems will relax by fluorescence, but some will relax by intersystem crossing to this triplet state. And basically just by a consequence of sort of the symmetry of these states as well as the dynamics of the system, you end up with a highly polarized electronic spin state just from this optical pumping process. This can be quite fast. And then you can transfer polarization from this electron spin state to your nuclei using classical DNP techniques like the integrated solid effect. And so, as I said, these are great because that polarization is independent of magnetic field and temperature. But the thing is, your source is trapped in a crystal and it's really far away from molecules in a solution or general molecules that you would want to polarize. So what happens is you can often, what a lot of really smart groups have found, you can polarize nuclei in those crystals extremely well, but then getting the polarization out is a huge challenge. And so Envision was first looking at MV centers in diamond, for example. This is still a really cool system, but we just kept running into a wall with not having enough surface and not being able to polarize really enough molecules to be able to see it. And of course, going to nano diamonds is definitely an option, but just the materials development needs more time, needs more work. And so we actually ended up focusing our strategy on the pentacene doped molecular crystals. We found really a lot of advantages there. So this was a huge effort led by my CTO, Eli Schwartz, my colleague, Tim, who's also here, who really specializes in this technology. So we brought this technology in-house. And so basically we decided on pentacene doped naphthalene crystals for a number of reasons. The first is that owing to a lot of really nice work from the Paul Scherrer Institute, from research groups in Japan, techniques for polarizing protons in naphthalene are now really well developed. So you can achieve up to 80% proton polarization in these crystals. The second thing is that they're very highly proton dense. So just a single crystal of naphthalene contains 71 molar protons. So this is just like a huge amount of magnetization that you can pack into a single crystal. The next thing is that this optically polarized electron spin state I mentioned with pentacene, it's actually short-lived, so it only lasts tens of microseconds. So what you can do is you can pump it, you can use it to transfer polarization. Once your nuclei are polarized, you can stop pumping, and then you have no more electron spins. And then this means that you've really minimized your relaxation sinks, and proton T1s can last days to weeks. So this enables you to actually polarize a crystal and then store it. And then you can completely decouple your polarization process from transferring it to something else. And then the final thing that is also really great about pentacene doped naphthalene crystals is that the naphthalene material is soft. So you can polarize your crystal and then you can really rapidly crush it or dissolve it. And so what this means is that you can use all of the advantages of polarizing something in a single crystal where polarization transfer is really efficient. And then you can manipulate it to maximize your surface area and really put the naphthalene molecules in contact with the molecule that you actually want to polarize. So another thing is the materials growth is also really advanced. So with the help of our chemists, so that's Michel and Sebastian, our material scientist, Martin, we were really able to also build the growth and manufacturing of these crystals from scratch up in Envision. So this is what we did. Okay, so now that I have a really nice hyperpolarized source, I want to use it to transfer to molecules in solution state. How does this actually work? So we've been calling this hypnosis. So it's a hyperpolarized NOE system. And basically what you do is you have your optically polarized crystal, you dissolve it in a system of target molecules, in a solution of target molecules. And then the polarization transfer from naphthalene to your target protons happens in the solution state. And so then finally, once you have your solution, you transfer it rapidly to your NMR spectrometer where you can either monitor polarization buildup or pulse and get your nice hyperpolarized spectrum. Okay, so then how does the polarization transfer actually work? And so we actually tried a few different implementations, but we found that in this method where you just dissolve the naphthalene in solution, you can actually get significant enhancements just through the intermolecular NOE effect. So this is an effect that we all know really well in the NMR community. It's quite a weak effect, but what we found is that because we can really concentrate naphthalene in the solution, we can just overwhelm our target molecules with magnetization, basically. So basically, we're just adding a huge amount of magnetization. And then with intermolecular NOE, the action of the molecules diffusing towards each other modulates the dipolar coupling between the protons on the naphthalene and the protons on the target molecule, enabling cross relaxation between the two. And some of you may know that these types of studies were done previously. So the Pines group especially, they looked at this type of transfer by bubbling hyperpolarized xenon into solution and then polarizing protons on benzene or partially deuterated benzene. And so they also saw enhancements there, but they were somewhat weak. And we think that our system is particularly suited because we're using protons and we're using high concentration versus xenon. So xenon has really large electron cloud, whereas protons don't. And so the nuclei can get closer. And also protons have a gyromagnetic ratio. So we just feel that the naphthalene system is really suited well to this. So as I showed you in the beginning, this is an example. So propargyl acetate is our benchmark molecule. We like it because it has different functional groups. So we were really curious how does this effect work with different functional groups? So the methyl group, the CH2 group, our SPCH group. And so at least with this molecule, we found that the enhancement really corresponded to the T1 of the different target sites. So longer T1 means more enhancement. So the highest enhancement we see is on the CH site, for example. And so we wanted to continue our studies of functional groups. So we also took really simple molecules like tetrachloroethane, dichloromethane, THF, acetone. So these all have different functional groups. And so with these molecules, we can get enhancements of 200 to 600 over 60 megahertz, 30 to 60 over 400 megahertz, and 30 to 40 over 600 megahertz. And what we also notice is, of course, other factors will certainly influence the polarization. So it's also really how exposed the protons are. And also we believe molecular affinity comes into play. So this is something that we're really starting to explore. The next thing I want to note is, I actually go back to one slide. You notice all of the spectra I'm showing here have actually had some processing, some background subtraction. And this sort of brings me to the next phase of my talk, which is the phenomenology. So something we learned is that when you are transferring polarization in this way, you're producing systems of highly polarized, highly concentrated naphthalene. So the radiation damping that we experience is extreme. And so what you're seeing in the hyperpolarized spectrum, so this is just a raw hyperpolarized spectrum, see a massively radiation damped naphthalene signal. And this is, of course, coming from the fact that if your magnetization is so large, when you pulse on your spins, you can produce a back action in your current, which actually gives you a magnetic field in the opposite direction, which pushes your spins back onto your z-axis, resulting in a really short FID, like shorter than normal, and a very damped spectrum. And so what we end up seeing is highly damped naphthalene spectrum, and then these narrow features added on top, which are our target resonances. And we see that they have an additional phase. And so basically, we can correct for this with a few phase corrections and also background subtraction to produce a nice, more conventional 1D NMR spectrum. The other thing we noticed is that because the magnetization is so large, all of the sample experiences actually a macroscopic magnetic field. And we see this actually in the target or in the whole spectrum. So what happens is the whole spectrum gets a shift, and then this shift decays exponentially over time. And so what I'm showing here are just spectra starting at early times and then going to later times over 100 seconds. And so you can see the target resonance grow and evolve and also shift over time. And so this shift that you see, the decay time is actually the naphthalene polarization loss time. So this is something that was interesting and also, of course, something that we have to handle when we're doing experiments. Okay, so this brings me to the last part of my talk, which is really what we're working on next. So of course, we can process the spectra and still produce nice 1D spectra. But what would be even better, if you're acquiring spectra where you still have this extremely large naphthalene signal, that means you have to reduce your receiver gain. So if you want to be able to acquire spectra where you can use increased receiver gain and really have maximum sensitivity, then source suppression or solvent suppression is sort of the next logical step. And so this is something we started to work on with our collaborators at EPFL, so Lyndon Emsley, as well as his postdocs and students, Penelope and Federico. And so we took this molecule, which we've been colloquially calling the phosphonate molecule, as to use to demonstrate this. And we like it because it has different proton functional groups. And it has different heteronuclei. And it was also chosen because it's sort of a safe version of a chemical weapons analog. So this phosphonate group and this fluorine, they're similar to sarin. So this was a nice molecule to study that would be similar to that. And so with this molecule on the protons, we get enhancements of 30 to 50 over 400 megahertz and 20 to 30 over 600 megahertz. So we get nice enhancement. And then here with our collaborators at EPFL, we also first use pulse sequence, your classic source suppression pulse sequences like excitation sculpting with perfect echo. And you can see that we can get nicely suppressed naphthalene signal and also be able to increase our receiver gain. So these are preliminary results and definitely something that we want to develop more with these guys. The last set of results that I want to show to you guys is our work looking at heteronuclei. So we still were able to use this phosphonate molecule to look at heteronuclei. And so we looked at both phosphorus and fluorine NMR. And so with phosphorus, what we did was we knew that we would get some enhancement to the protons. And then we thought we would use the target protons actually to transfer polarization to phosphorus. So we just use a classic inept sequence. And so this spectrum here is comparing a thermal inept to our hyperpolarized inept. And at 400 megahertz, we get an enhancement of 8 on the phosphorus. And then finally, for fluorine, we actually just tried to see if we could get direct transfer to fluorine from the hyperpolarized naphthalene protons to the fluorine nuclei. And there we've seen that we can already get an enhancement of 4 over 400 megahertz. Okay, so just to conclude, we've shown that we really can use optically polarized sources to get nice hyperpolarized solution state spectra of small molecules. The technique that we use, which is just solution state transferred by intermolecular NOE, produces enormous magnetization, which gives interesting radiation damping and added phase effects. And then finally, we started to look into source suppression now in heteronuclei, and we're really excited about trying other things. Finally, of course, I want to thank our team. So Envision has an amazing team. It's really interdisciplinary. We have people from all over the place. We have physicists, chemists, material scientists, and the work that I showed you is really like a culmination of hard work over several years. And so I really want to thank everybody on this team. I hesitate to especially acknowledge people because so many people really, everybody really contributed well, but I do want to acknowledge Tim Eichhorn and Eli Schwartz for really leading this project and building up, leading how we built up this technology with pentacene doped naphthalene crystals in the company. And then I also want to thank John Blanchard and Stefan Knecht for some really instrumental ideas and understanding how this works, and also for translating this to high field, and personally for mentorship. And then, of course, we have our co-founders and advisors at University of Ulm, Fedor and Martin, who are really great. Our collaborators at EPFL, Lyndon, Penelope Federico, also at PSI, Patrick, Aoife, and Jakob, were extremely helpful in getting this up and running. And then also recently, work with our colleagues at Bruker has helped us also get to next steps. And then finally, for those of you, so Envision is also really interested in hyperpolarized MRI. If you want to see any of our developments there, definitely check out this talk by my wonderful colleague, Alistair Marshall, who will be presenting at 11 today. So thank you all very much for coming to my talk. Thank you for your time.
Questions for?
Yes.
That is such an exciting development.
Thank you.
I was wondering, the obvious question is, does the intermolecular NOE work better at low field or at zero field?
So it really depends on the molecule. So also our collaborators at EPFL are doing simulations. So we're finding that low field can be definitely a really nice regime to work at. So this is something that we want to look at more. Yeah.
And can I ask a follow-up?
Of course.
So there were so many authors on this work. Whose actual idea was it to dissolve the pentacene crystals? It's really ingenious.
I don't even know at this point. Do you know, Tim? Yeah, I think this idea was had by a few people, at least. Yeah, sorry.
Yeah, very nice talk. So I have actually maybe a dumb question. This is just one scan thing, is that right?
Yes. Yeah.
Okay. So, and also related. So if I actually So what would be the dead time for running this kind of experiment? I have to really throw in the solution, how long I have to wait to make sure it's dissolved and transfer the spectrometer? How long this typically takes?
So let's see, the dissolution process takes 6 to 8 seconds, transfer takes a few seconds.
So
So it all happens in less than a minute. And I will say, so something that we would like to see is, the damping effect is possibly helpful. So the radiation damping can actually push the magnetization back to the Z-axis. And so then we wonder if we can actually, have another buildup of an intermolecular NOE in solution and get more shots. So this is something that we're looking into.
okay.
So possibly you could get more, but of course we have to study it.
So these are all just done at room temperature so far,
Yes.
right? So if I simply just bump up to like elevated temperature for some like hard to dissolve systems,
This is also something we want to do. Yeah, definitely.
okay, but that, I guess the enhancement will somehow will change correspondingly.
Yeah,
to
yeah.
melody. Okay, all right.
Absolutely. Yeah.
Thank you.
Thank you.
Yeah, thank you for sharing this. Really fascinating. My question is, if you're having this radiation damping effect, is that interfering with the pulse sequences you use, like the INAPT, or can you solve that? Or how do you solve that?
So we believe that for, at least from what we know currently, we believe it does affect it for the naphthalene, but at least for the inept, we're hoping, so we don't see that damping on the protons on the target molecules. And so at least from what we can tell, it is affected on the naphthalene, but then since the actual resonances on the target molecules aren't damped, and that's where we want to transfer polarization from to the heteronuclei, then we don't think it's affecting it, but that's just what we think so far.
Okay, thank you.
Thank you.
Hi, thanks for the nice talk. I'm curious, does the T1 stay long after you crush it? I mean, if you have a powder, can you leave it there for a while you do a series of experiments?
So with the powders, you definitely get a reduction in T1. And if you can keep it cold, that certainly helps. So I think with powders, we've observed T1, like if you keep it cold, I think you can get T1s for like 30 minutes. But then if you're at room temperature, it can be like 5 minutes or maybe worse, actually. So when we dissolve it, yeah, we try to dissolve it as fast as possible. And then in solution state, the naphthalene T1, at 1 Tesla is about 20 seconds. At half Tesla, it's still about 10 seconds. And then when we inject a high field, actually, we get long T1s again, like 25 seconds. Yeah.
Okay, thanks.
Go ahead, Chris.
Sorry, very nice work.
Thank
Going
you.
back to the crushed sample, did
Yes.
you try to mix it very intimately with the powder and spin at the magic angle?
We haven't tried that. I guess we were always worried that we wouldn't have enough time to properly do it, like crush it and get it into a rotor. But we're really happy to try things.
And you're working at room temperature, am I correct?
When we do the crushing and dissolution, it's all at room temperature. So we also want to try like different temperatures, but all the work was at room temperature.
What are the range of chemical identities that you can change about the naphthalene and the pentacene in that system? I mean, one change simple change that you could imagine making is to deuterate selectively and symmetrically on the naphthalene to narrow the spectrum so that it has only a single line in it, for example. But other polyaromatics and so on.
Yeah, I think it's a great question. I think so. The Japanese groups who work with these types of systems have really looked into this a lot. So I think there's a lot you can do there, and they've also looked at functionalizing pentacene for different purposes. Also selective deuteration or full deuteration, stuff like this. They've absolutely looked into. I think PSI has also done a lot of work here. So I can
published.
Yeah, so if we meet afterwards, I can get your email and send you some publications or something.
Thank you.
Last brief question, please.
Okay, I'll try to keep it brief. So I have two questions about your radiation damping or your distant dipolar shifts. Do you account for the shift while acquiring your FID because the field will change during that?
Good question. Yeah.
You do?
No, it's a good question. We want to, but we haven't done that yet.
Okay,
No.
and the
So we
second
sometimes see broadening from that. Yep.
Exactly. And the second question, do your phase shifts, are they the same for all groups you polarize?
Yes. So the phase shift we believe is coming from the radiation damping field of the naphthalene. So yeah, you see a consistent phase shift on the different resonances, and then that changes exponentially over time. So you see it sort of often start around like 70 degrees, and then it will evolve to 180 degrees, which is like what you would expect from an NOE effect.
Thank you so much.
No problem.
In the interest of time, we will move on. Thank you, Anna, for
Thank
your
you.
presentation. Next is Ashok Ajoy from UC Berkeley.
I need your help, actually. That's fine. I can just mouse. This mouse will work. Okay. Excellent. Okay, excellent. Well, thanks for the opportunity to be here. So today I'm going to tell you about some new work we've been doing, also with optical hyperpolarization, slightly different, actually trying to make sensors out of polarized nuclei. And so this is work from my group at Berkeley. So we're going to work on this system, which is hyperpolarized carbon-13 nuclei in diamond. So the process we use actually is similar to what Anna was saying. These are electrons, the NV centers in diamond. They can be very easily hyperpolarized by shining light. And then you can transfer this polarization to the C13 nuclei. So this is old work, and I won't go into it in much detail, but basically this is the protocol we employ. This is laser pumping for a few seconds, followed by, or in conjunction with chirped microwave radiation. This is done at low field. And then when you carry the sample up into high field, you can very easily see quite a large, significant polarization developed on the C13 nuclei. The polarization levels we get are typically few percent, and it develops in under a minute. Actually, in today's talk, I'm going to focus on some kind of interesting properties that we got after we polarized nuclei. So, these carbon-13 nuclei can be so easily polarized, but then once you polarize them, you can sort of unlock their kind of interesting properties. And one of the properties is that quite remarkable coherence times and things that you can do with this long coherence. So let's consider a simple experiment. So if you were to simply do an FID, which is pi half and a quarter, you'll find that the decay time of this is about 1.5 milliseconds. And this is given because the carbon-13 nuclei are dipole coupled to each other. On the other hand, however, if you were to be a little bit more clever and do something like a spin lock, and I'll describe in a moment, you can extend this quite significantly. So the fact that you can extend FID lifetimes is not a surprise, but the surprise was the extent to which you can extend it. So we were able to go at room temperature to more than 90 seconds. And you can see the FID going to more than to about 10 minutes in total. And what we're doing in this experiment is we're doing a pulsed version of a spin lock. So it's a pi half followed by a train of theta pulses, so theta, 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 etc. And then we are measuring in between the pulses here. And that's what each of these windows looks like. So very high signal to noise. So you can see really the Larmor precession of these C13 nuclei. And then you can collect all of this and produce a graph like this in one shot. So this experiment is interesting in the context of the total number of pulses you can apply. So it's a very kind of forgiving system. So we apply close to 6 million pulses. We have 6 million windows. So this graph actually has 6 million points. There's a little data handling problem, but then after that, you can produce a graph like this, which, essentially you can lock the spins along the x-axis around some transverse axis for a very long amount of time. So today I'm going to tell you about how we can exploit this to do some interesting things. So I'm going to tell you two short stories. First, how you can use these long transverse spins as sensors. And second, maybe how you can make designer spin trajectories and manipulate where the spins live in the Bloch sphere and track them over time. On this first story, actually, we were motivated by some old ideas, which is basically, can you use hyperpolarized spins not as polarizing agents like we heard in the last talk, but instead as sensors of nuclear spins that are external to these polarized nuclei. So let's consider a thought experiment. A typical NMR involves the detection of weak oscillating or time-varying magnetic fields from analyte nuclei, which you pick up with an RF coil. So these fields are very small typically. So, for proton in water, for instance, in a 10 micron cube volume, the field that it produces about 10 or 11 nanotesla. So, you can think of NMR as some sort of magnetometry that's carried out at high fields. On the other hand, if you consider polarized nuclei, they have a much higher signal to noise, so they can be much more easily, or they have a much higher polarization, so they can be much more easily detected. So the experiment that we're trying to set up is this sort of relayed process where you take these hyperpolarized nuclei in some kind of nanoparticle, so it's spatially confined. You bring it in close proximity to your analyte nuclei. So the analyte nuclei NMR signal is encoded in the polarization of these hyperpolarized nuclei that is then picked up by the RF coil. So it's a relayed process. So the hyperpolarized nuclei in this spatially confined volume act as a transformer or an amplifier to increase the signal that you would get from these analyte nuclei. And this only works because you're basically able to measure signals in a spatially proximal to these polarized nuclei. And this is similar to what people do, for instance, in zero field NMR. So the rubidium sensor people use in zero field NMR has a similar concept. So today I'm going to tell you about some steps towards this direction. And what we've been trying to do is make these C13 nuclei as sensitive magnetometers at high fields, so you can measure very weak time-varying magnetic fields with them. It's motivated by the fact that the spin sensing in general, it goes by the name quantum sensing. It's just jargon, but there are many ways by which you can make these spin sensors. A popular sensor is the NV center in diamond, where once again you've got an electron, but nuclei around it, and you try to measure the presence of these nuclei by measuring the weak fields produced by the nuclei on the electron. And you can measure chemical shifts, for instance. The disadvantage of these methodologies is that they use electrons and they have to be limited to work at low magnetic field. This is also the case for rubidium, where that's an electron as well. And so you have to work at low magnetic fields. And when you work at low fields, basically you lose chemical shift information, or it's very hard to discriminate chemical shifts. So instead, our goal is to make these C13 nuclei sensors with the big advantage that they can operate at high fields. They have some other advantages. They are, compared to NV centers, they're more abundant. They are spin half and they're independent of orientation, and they have very good coherence properties. So very long T2 and T1, and they can be polarized. And then one can envision doing interesting things, for instance, taking a micro droplet, putting nanodiamonds inside them and using the nanodiamonds as sensors of chemistry happening inside the droplet. So you use it as a spatially proximal NMR sensor with a length scale target of about 1 to 10 microns. So that's so we're trying to do micron scale NMR with them. So let me show you just how this will work very quickly. So let's consider the experiment that I just showed you, where you do this pulse spin locking. And essentially what happens is the C13 nuclei are locked along one axis. I'm going to call this axis X. And that's the typical signal that you get going to about 20 seconds. So very long spin locking lifetime. Now, if you assume that there is a weak magnetic field along the Z direction that these spins are subject to, then you find that if you were to look at that signal once again, you find that it's on the signal is riding actually very strong oscillations. And so it looks like this blue line here. This blue line looks like a band because there are actually oscillations in them. And what's going to happen is you can think of the spins as undergoing a secondary precession. So this thing that was locked along the x-axis now undergoes a secondary precession in the rotating frame in the YZ plane. And because it precesses along the YZ plane, you can find basically oscillations in its signal amplitude. So this is what that data looks like if you blow up into it. So this is the data for 20 seconds. But if you blow up into a small window, you basically get very coherent oscillations. Yeah. So this is a test experiment with a synthesized AC field. We're applying a very small time-varying field of the order of a few tens of nanotesla, and that's the signal that it produces. If you then do a Fourier transform, you can basically extract all the components of this field that you put in, its amplitude and its frequency, with very high resolution. The resolution here is determined by the total length of time for which you can measure. So essentially what's happening is that these C13 nuclei have then been turned into magnetometers or magnetic field detectors. They work at high field, but they're still able to measure very weak time-varying magnetic fields. And what ongoing work in my lab now is trying to use this to detect NMR at short length scale, so 1 to 10 micron. let me just show you just as a technology comparison where this magnetometer would lie compared to other technologies. So on the y-axis of this chart is sensitivity. The x-axis is bias field. And you can see that essentially for NMR detection, what you want to have is high sensitivity and high bias field. So you want to sit on the right corner of this graph here. But already this sort of proof of concept experiments that we performed shows a sensitivity that's below 1 nanotesla per root hertz, and significantly better than NMR, conventional NMR magnetometers, but also better than NV centers in this regime. So we hope to be able to make micron scale NMR detectors with this idea. So let me move on. So this work actually gave on to some more interesting cool things that you can do with these spins, just because you can lock them for so long and you can measure them continuously. And so we started trying to see whether we can track the dynamics of the spins in real time. And this was a surprise. This is very new data. It's like literally like a few weeks old. And it took us a long time to figure out what's happening, but let me show you what's up. So this is the data that I showed you. So this is a long time spin precession, and you can see it out here and it's zoomed in. Actually, if you were to do this experiment more carefully, then you can find that these oscillations can be preserved for very long times. So this was a surprise. So this shows the signal as before here, but instead of just measuring amplitude, we can measure amplitude and phase. So this is amplitude and phase, and this is for a signal around 5 seconds and around 15 seconds. So it's a 5 millisecond window. So 5 millisecond, around 5 seconds and 15 seconds. You can see that you have these really coherent oscillations, and it's so coherent that you can almost connect every fourth point here, and you can see that they all fall like in straight lines. Yeah, and I'll show you why they fall in the straight line in a moment. But essentially, these oscillations of the spins in this YZ plane on the Bloch sphere continue for a very long time. So what we started to ask is whether we can extract the three-dimensional position of the spins in real time. And, this is usually an impossible thing because you can only measure amplitude and phase, so you can only get information about the XY components of the spin. But then you can use the fact that you're measuring so fast and the spins have such a long lifetime that they hardly decay, and use a unitarity constraint to also estimate the Z component. So the Z component has some error in the estimation, but you can do a pretty good job of estimating X, Y, and Z positions of the spin in real time. So you can almost track the trajectories of the spin in real time. That's what it looks like. So this thing that I was showing you as a theoretical simulation now looks like this. This is real data where you can see in this small window what the positions of what the spins are actually doing. So they're undergoing this trajectory on the YZ plane here, and these are its two projections on X, Y, and YZ. Yes, it's slightly tilted, but this is a technicality. So then we started doing some interesting things with it. So we asked, can we track the position of the spins and make the spins go in some weird trajectory that you can decide? And so this is all real experimental data I'm going to show you. It just looks like somebody's made cartoons, but this is real data. So this is the experiment that's shown here. So we have a pi-half pulse and a train of spin locking pi-half pulses. And an AC field is applied that's exactly periodic with 4 pulses. And if you then plot the trajectory of the spins in the YZ plane, so this is YZ, and time on the Y-axis, on the vertical axis. So, the position of the spins is in 3 dimensions, so it's hard to plot something in 4 dimensions. So I'm going to plot the position of the spins in the YZ plane, and then I'm going to use time as the vertical axis. You can see that the spins basically make this trajectory that's exactly a square. So if you extend it out, it looks like a perfect prism. And so we have tracked here the position of the spins in real time, and we do this for basically 30 or 40 seconds, and it continues like this for a very long time, which is a surprise because you'd expect the spins not to last so long. Yeah? But this prism lasts for a very long time, and I'll get to in a moment why this is the case. And then you can be more creative. So you can do, for my students who are learning NMR, decided to play tricks with spins. And so you can make the spins go in a trajectory that looks like a pentagon, a star, and an 8-legged star. And this is all tracked in real time. So, so it goes to show that the system is so forgiving and so kind of coherent that you can make it do whatever, and you can also watch it as it does it. So then we started playing more interesting games with it. So this is again real-time tracking of the spin trajectories, YZ plane. And what we're doing is we're changing the phase of this field that we have applied. And you can see that this basically the trajectories turn as you'd expect. This is all one-shot data, so there's no averaging, so very high signal to noise that we have in these polarized spins, and you can make these spins go in a trajectory that you predict here. And note the time axis. So this is 7 seconds into the trajectory, but it's still doing this motion that looks very, very stable. And then you can ask, how does this trajectory emerge? So here's an interesting thought experiment. Say I apply a perfect spin locking pulse. So the spins are locked along X. I'm going to denote it by this line here. And then I'm going to apply this AC field. So the spins now suddenly go in a motion that looks like a square. And then I'm going to turn off this AC field, okay? And sort of collapse back into something that's spin locked. And if you track the motion of the spins in real time, this is the trajectory that you get. So once again, tracked over a long time, but really beautiful and somewhat surprising. First time we saw it, it's like somewhat unexpected that it should work so well. So so this is spin locked. So that's why in YZ it looks like a line. So it's a perfectly spin locked. And here the student opens this AC field. You can see the spins like trying to equilibrate, and finally they equilibrate into this thing that's a square. It goes on for a long time until they stop, and then it collapses back into a line. And this is one-shot data again, so very, very good SNR. So you can play more games with this. So this is again increasing the strength of the field. So you can make on the YZ plane something that looks like a thin cuboid or something that looks bigger. An interesting thing is even if you make it pretty big, the decay time does not change. So it still remains very, very straight for seconds together. Yeah? And we see this motion going for about 30 seconds with very less decay. And then what you can do is you can do this experiment in interesting configuration, which is you make a variable square and you collapse into a line and you make a fixed square. And this is a movie that shows this. So this experiment is basically you've got a AC field is applied, you're varying the size, strength of this AC field. Then you switch off the AC field and the spins will collapse back into a spin locked axis. And then you have one for comparison here that's fixed. You see that even if you increase the size of this thing, this does not decay. So essentially the message is you're able to preserve the trajectory of the spins on not on the spin locked axis, but on some other axis, but you're still able to preserve it for a very long time. And that's an interesting surprise. So finally, let me show you some more things you can do with this. So in this experiment, my student has made a pentagon in the same kind of experiment. So you start with spin lock and then you open a pentagon. The spins make a pentagon and then you wait for some time and you make a square. And then you can make this pentagon and square collide with each other. Yeah. As you can see, they're having fun with spin dynamics, so they're learning. But you can see here the real-time track data in one shot of this these spins doing this. So this is, again, note the time axis is 7 seconds. So this pentagon lives for 7 seconds. And then you lock the pentagon into a spin lock, and then you open a square. Then you can vary the time here, and you can see them colliding with each other. And then they will finally basically completely collide into each other, and you can see something that looks complicated. But when you open it up, it's again a pentagon and a square. And here's a movie of this happening. So the phases every time we do it are random, so that's why you see it like doing that. But you can see it happening. So anyway, so what is the goal of all this? So right now it's still like a fundamental curiosity, I should admit. So it's sort of interesting that the spin lifetimes can be preserved for a very long time. The advantage is because they are so highly polarized and the couplings are relatively weak, you can really manipulate the trajectories and be able to watch them in real time. What we think it's useful for is to make like new types of spin sensors that exploit the symmetry of these trajectories. Because you know that the spin goes in a fixed trajectory, any field that you have that changes the symmetry of this trajectory can be detected very much more sensitively than if you were just measuring deviations from a single axis. So that's what we're doing next. So let me conclude by roughly just telling you what I did here. So we try to show that you can make these spins as sensors of weak fields, but also interestingly, you can track the evolution of the spins under these fields in real time for quite long times. Let me acknowledge all the students who were involved in this project. So many smart and young students and collaborators, Deidre Suter, Ben Gilbert, and Marine Buchhoff helped with the theory. So thank you.
Questions for Ashok? Amrit.
Oh, could I, just before you start, I should say, actually, there's a postdoc position open in my group, so if anyone is interested.
Yeah, thanks for that really cool talk. I have a very naive question. So thinking about your carbon spin locking and the polarization, long-lived T2s or T1 rows, however you'd like to call it. I'm trying to relate that with solid state. There are two types of cases in solid state NMR. We see this type of thing, either in cases where you have inhomogeneously broadened signals, your refocused T2s are longer. Alternatively, you have homogeneously, like dipolar broadened signals or something like that, and then you have sort of a homonuclear decoupling effect, consequently leading to, longer T2s. So I can't see this case relating with either.
Yeah, so this is certainly the broadening is homogeneous broadening. So the inhomogeneous part is weaker, but the mechanism is not decoupling. So in the normal spin lock, the mechanism can be just thought of as how spin lock works. You're truncating the Hamiltonian to commute with the spin locking axis. So you only preserve spin states along this axis. So that's easy relatively. The more interesting thing is how can you take the spins out of a spin lock axis and still preserve the trajectory for a long time? And now we understand why it's working. So we think actually what's happening is we are exciting dipolar order. This dipolar order survives for a very long time. So actually you're making a state that's thermalized with the dipole Hamiltonian plus a single magnetization term. And this can be preserved for a very long time.
And was it carbon labeled? Is that right?
No, this is 1% natural abundance.
Okay, interesting. Thank you.
John.
Lots of different kinds. For the transducer part, in what regime do you expect to actually have a gain greater than 1?
Yeah, so you'll get a gain rate. If you're trying to measure nuclei that are spatially proximal, so small nuclei with a big coil, that's when you'll get a big gain, right? So normally in NMR, it's hard to make a coil that's 1 micron in size. But say you've got a nanodiamond, which is 1 micron in size, and you go in close proximity with something, then you can only measure stuff which is about 1 micron. But you can still get a gain because the signal is amplified by the polarization of this agent.
So are you able to distinguish between local versus bulk spins? Or
No,
just
so
for very small
the point spread function of the sensor will be given by the size of the particle that you use. So if you use an ensemble of about 10 micron, then you can detect about 10 micron. It's the same approximation that's we used in rubidium cell sensors.
Thank you.
Lucia.
Thanks, Ashok. Amazing stuff. I want to go back to this first question that you were asked about how can it be such a long lived of so many tens of minutes? If these are natural abundance diamonds, I don't know, maybe you mentioned if they are single crystals or not, but they have a dipolar broadening of
1 kilohertz.
Right? Yeah,
right.
Exactly.
Exactly.
So, and you somehow have to preserve this millisecond lifetime over 10 minutes, which is, so is this related to the hyperpolarization? Is it because if it's a single crystal? Is it because of, well, what preserves, I mean, even the dipolar, have you measured the T1D?
No, we haven't measured T1D. The T1 is very long, so maybe half an hour. T1 row is this 100 seconds or so, is the approximately. And yeah, the T2 star, which is the FID, is 1 millisecond, right? So it's very fast. Yeah, so I mean, if you think of T1 row, you would expect that to be pretty long, so that's not surprising. And T1 row has a long way to go before it hits T1, so 100 seconds maybe is not surprising. What was surprising is when you take it out of the spin-locked axis, you would expect that the spin locking will not work anymore and this will die. And yeah, this took a long time, I think. So I've got some theory on it, but I'll skip the details. But ultimately, it turns out that that is the Hamiltonian that you engineer. It looks something like this. V's are just like single particle operators, but you basically get the dipolar Hamiltonian plus a single magnetization term. And what we think is happening is the spins are thermalizing to this Hamiltonian. So essentially exciting dipolar order. And for some reason, again, the dipolar order is so long-lived. And what we're measuring is only so basically when it thermalizes, the density matrix looks exactly analogous to this Hamiltonian. And so then you're only measuring this term, and this is the term that you manipulate with all the pulses.
Maybe the T1D is also several minutes.
Yeah, and interestingly, why it's so long, yeah.
One last question.
To ask you, do you have to optimize like the spacing between the pulses?
No, it actually works very well. There's a reason why it's easy to do, right? So nothing, you just dial in the number. All you need to do is for making these shapes, which are all like regular polygons, you have to make this period of this field exactly equal to like some number of pulses. That's it. But After that, you can just make these shapes and they survive for a very long time.
And in the first slide, it looked that you are not even on resonance with.
Which one?
In the very first slide.
Yeah, so that's, so there you're trying to detect a field that's unknown. So here we're actually using the same principle, but you can drive the trajectories of the spins by now, instead of sensing it, you're actually driving it with this extra field that you apply. Yeah, so this is a spin lock in the X and a Z field, a weak Z field that drives this motion.
With that, let's thank Ashok one more time. It's funny to hear this kind of talk and several that we've heard that are usually in Exotica, but now it's in the materials section. So that's a neat thing. All right, our last speaker of the session is Ken Harris from Cardiff, who's joining us online from Zoom. So you can go ahead and start.
Okay, so can you hear me okay?
Yes, we can.
That's great, thank you very much. So I'd like to begin by thanking the organizers for the kind invitation to give this lecture. I'm very sorry not to be in Orlando in person on this occasion. But what I'd like to do during this lecture is to discuss some of our research over a few years now on developing in situ methods to explore crystallization processes. So I'll begin with some background to the basic idea. And basically the idea is to monitor the evolution of the solid form as a function of time during the crystallization process. And let me just illustrate schematically the sort of process of interest. As I'm sure you know, crystallization, basically the free energy barrier that one must overcome in crystallization is to establish the critical nucleation entity. So the first stage is called nucleation, and once nucleation has taken place, then simply those nuclei will grow into larger crystals. So what I've shown there is the simplest situation, but this is not always what happens, because what can often arise is that nucleation gives a metastable form of the solid material, and then subsequent transformations occur during crystallization to produce a more stable solid form, a more stable polymorph. And this could actually go through a sequence of several different polymorphic transformations before the final crystallization product is obtained. And what we want to do then, using the in situ NMR techniques, is to interrogate all of these stages of the process to identify the sequence of solid forms obtained during crystallization. And clearly, as I'm sure you all know, high resolution solid state carbon NMR is a very powerful method for distinguishing different polymorphs of organic molecules, illustrated here for glycine, which is a very well widely studied polymorphic system. 3 known polymorphs, alpha, beta, gamma, structure shown on the right-hand side, but the carboxylate carbon chemical shape is clearly distinct for those different polymorphic forms. Okay, so let's now consider doing this in situ in a solid state NMR spectrometer. The idea is very, very simple. We carry out crystallization in the NMR spectrometer. In general, we induce crystallization by rapidly decreasing temperature and then simply measuring the solid state NMR spectra repeatedly as a function of time during the crystallization process. And schematically, this is what we're doing. We start with an undersaturated solution at high temperature, cool rapidly, and we simply allow the system to evolve, measuring the NMR spectrum as we go. And here we can see the nucleation of growth of one polymorph monitored from the solid state NMR spectrum, but then we may get a polymorphic transformation as shown here, which again is identified from the changes occurring in the solid state NMR spectrum. But first of all, the question is why use solid state NMR? There are many spectroscopic techniques, diffraction techniques, that can be used very productively to look in situ at crystallization processes. But what we recognize here, and this was the motivation to begin this work, was that NMR has a big advantage that we can set up the experiment to selectively detect only the solid phase. So let's suppose only 1% of the solute is crystallized. There's a tiny amount of the solid phase present, but we can set up the measurement in such a way that we detect selectively only that component, and the liquid phase is invisible to the measurement. So one way we can do this, and I'm sure this is very basic, one way of doing this is simply to use cross-polarization. Here's a heterogeneous mixture of glycine and water, and it's a doubly labeled glycine, and measurement under normal direct polarization gives a spectrum of this type. Zooming in on this particular resonance for the carboxylate carbon, we can see the signal from the solid phase, but superimposed on that's a huge signal from the dissolved glycine, but under cross-polarization, we selectively detect the solid phase. Now, clearly in this time-resolved study of crystallization, time resolution depends on the time to record a single solid state NMR spectrum of adequate quality to identify the solid forms that are present. And sensitivity undoubtedly is a big issue. So we might want to consider isotopic labeling, and in particular to carry out measurements of high magnetic field. And virtually all of the work I'll describe today is carried out at the UK High Field Solid State NMR facility at the University of Warwick. And we're very grateful to them for a lot of time for these experiments. Okay, let's begin by looking at some examples of polymorphic evolution probed using the in situ NMR technique. So our first successful measurements were a few years ago now, and I'll return again to glycine. I should emphasize here 3 polymorphs, as I said, but the order of stability, gamma is the most stable, alpha is the next stable, and beta is pretty unstable and rather hard to obtain. So anyway, crystallization of glycine from water is well known to produce the alpha polymorph. And that was our first measurement. You can see here the carboxylate region of the carbon NMR spectrum recorded over a period of time during crystallization. We see the formation and the growth of the alpha polymorph. But when we then looked at the same process in deuterated water, we can see quite a different behavior. Now, at the beginning of the crystallization, the nucleation stage proceeds in exactly the same way as in natural abundance water, but then after about 2 hours after beginning the crystallization, we can see a polymorphic transformation from the alpha polymorph to the more stable gamma polymorph of glycine. And if we quantify these measurements, we need to calibrate the cross-polarization efficiencies of the different polymorphs. You can see very clearly here the initial rise of the alpha polymorph, and then after about 2 hours after beginning the crystallization, the alpha polymorph decays, and that is mirrored by the growth of the more stable gamma polymorph. So this is the type of polymorphic transformation of interest to us. If we now crystallize glycine from a methanol water solution, we can see a different behavior. In the very early stages, we form the very unstable beta polymorph, and then it transforms to the alpha polymorph, as shown there. And what this really gives us is the information in the time window during which individual solid state forms, individual polymorphic forms are present during the crystallization process. So, for example, obtaining a pure phase of the beta polymorph is not an easy thing to do. And what we can learn from this experiment is that if you want to obtain a pure phase of the beta polymorph, we need to begin the crystallization and then stop it immediately. Because at the very, very beginning of this crystallization, we actually have, as you can see here, essentially a pure phase of the beta polymorph. If you run the crystallization for any length of time, it is already a mixture of both the beta and the alpha polymorphs. So there's just a summary of the work there I described on glycine crystallization. So glycine is already a very well characterized polymorphic system, but there's also the possibility through these measurements to discover new polymorphic forms. I want to give one example of this concerning co-crystals of dihydroxydecane and urea. Now, these form part of a much wider family of hydrogen-bonded co-crystals, and here is the in situ NMR experiment using carbon-labeled urea. So we're focusing on the urea signal in this in situ measurement. What we can see initially, we can see a signal at this position, and then quite suddenly, after about 40 minutes of the crystallization, that transforms to another much stronger signal, which corresponds to the known, there's only one known polymorph of this co-crystal. So clearly what we have at the beginning is a new transient polymorph, and from the information from the in situ study, this tells us there is the existence of a new polymorph, and moreover, it tells us a time window within which we could stop the crystallization, isolate that polymorph, and then subject it to various structural studies for structural characterization. So characterization of the time depends of the solid form present during the crystallization process. So what I just described to you was the initial type of experiment, and then we realized there's actually a much better way to do this experiment, a much more informative way. And this is what we call classic NMR. Now, classic doesn't mean old. Classic means combined liquid and solid state in situ crystallization NMR. The point is that during crystallization, we're focusing in the previous experiments on the solid form, the evolution, the formation of the solid form, but at the same time, as a solid form is obtained, there are also significant changes occurring in the liquid phase, in the solution phase. And to gain a complete picture of the crystallization process, we actually need to be able to characterize that aspect of the crystallization system as well. So basically what we do in classic NMR is to use two different pulse sequences, one to measure the solid state NMR spectrum as before, and one to measure the liquid state NMR spectrum at the same time in the same experiment. So this is the strategy here. We have two different pulse sequences. One might be cross-polarization, as described by before, to measure the solid state NMR spectrum. The other might be direct polarization with short recycle delay, or maybe without proton decoupling, to measure the liquid state NMR spectrum selectively. So we alternate between two different pulse sequences during the crystallization experiment, and then at the end of the experiment, we separate these into two sets of time-resolved spectra, one set of time-resolved solid state NMR spectra, and one set of time-resolved liquid state NMR spectra. And the information we obtain, for example, for the solid state, is similar to what I described earlier: changes in the amount of the solid phase as a function of time, changes in the solid form as a function of time, and in the liquid state, information like change in concentration with time, change in solution state interaction with speciation, and so on. Okay, so let me give you one example of the application of classic NMR, and I'll focus here on crystallization of meta-aminobenzoic acid from DMSO. Meta-aminobenzoic acid has 5 known polymorphs, as shown there, and they're clearly distinguished on the basis of their solid state NMR spectra. Here's the solid state component of the classic NMR data. What we can see in this experiment, there's actually a delay time of around 2 hours where there's no signal in the solid state NMRs. There's an induction time before nucleation happens. This is quite common in crystallization. You know, nucleation is a rare event, stochastic event, and in this case, the solution had to wait for around 2 hours before the right critical nucleation cluster arose before crystallization would then proceed. So this is quite important in our subsequent discussion, is induction time before crystallization begins. In this particular case, we get form 1. And I should emphasize, actually, let's go back to this. Some of the polymorphs are zwitterionic, others are non-zwitterionic, and that's going to be important in a moment. But in this particular experiment, we produce polymorph 1, which is known to be zwitterionic. There's actually no polymorphic evolution in the solid form during the experiment. Looking at the total integrals of the solid state and liquid NMR data, we can see no change in the amount of the solid, no presence of the solid in the first 2 hours. We only have the liquid. And then after 2 hours, we see the increase in the amount of the solid mirrored by a decrease in the amount of the methylamine benzoic acid in the solution phase. But much more informative in terms of the liquid phase is to look in detail at how the chemical shifts in the liquid phase change as a function of time during the crystallization. What we're going to do is to look in, we're going to zoom in to the change in the chemical shift for all the 7 carbon environments in the molecule, as well as in the DMSO solvent as shown here. So let's look first of all at carbon number 6. I'm going to show all of these results on the same scale of 0.5 ppm. So in the first 2 hours, when nothing is actually happening, the chemical shift is constant. And then, as soon as crystallization begins, the solution state chemical shift changes in a very well-defined way. If we look now at C3, the same thing happens, but in the opposite direction. No change in the first 2 hours. And then once crystallization begins, once crystallization proceeds, there's now significant change in that chemical shift. And the same arises in its own characteristic way for all the carbon environments in the molecule. And even the solvent, as you can see here, the chemical shift changes in a very well-defined way as a function of time once crystallization is actually proceeding. So how do we interpret all of this information? So what I'm doing here is putting all of these data on one graph where I'm expressing the carbon chemical shift relative to the value at time 0. So this is the relative change in carbon chemical shift as a function of time. So in the first 2 hours, when nothing is actually happening in the crystallization, there's no change in the chemical shifts. And then as soon as crystals are formed, and then as crystal growth proceeds, we see very well-defined changes in all the liquid phase chemical shifts. Okay, so what we have at the end of the crystallization experiment is an equilibrium saturated solution. What we have at the beginning in this 2 hour window is what we call a pre-nucleation supersaturated solution. That's a thermodynamically unstable solution because it wants to crystallize, but it's just trying to find the right mechanism, the right nucleation pathway to allow the crystallization to happen. And this represents a decrease in concentration, as I described earlier. So the way we interpret this data is as follows. At the end, we've got an equilibrium saturated solution, and that can be studied independently by NMR and other techniques. So we can get a very complete characterization of that solution. And what we're going to do is to rationalize all these changes in carbon chemical shifts, looking backwards in time to give us insights into the nature of this thermodynamically unstable solution before nucleation actually happens. And here's just a very, very brief summary of the conclusions from this type of analysis. So what we know is at the end of the experiment, we get solid NADA form 1, and this is a zwitterionic form, a zwitterionic solid form. We know that in the equilibrium saturated solution, in the liquid phase, the molecules are non-zwitterionic. And if we rationalize all of these changes in chemical shifts, looking back in time in the liquid phase, what we deduce is that the pre-nucleation supersaturated solution relative to the final equilibrium solution has an increased proportion of zwitterines or non-zwitterines engaged in OH-N hydrogen bonding. And both of these scenarios described here are very plausible in terms of the type of precursor that may exist prior to crystallization in the supersaturated solution. So I want to move on quickly to the final two sections. First of all, detection of amorphous intermediate phases on crystallization pathways, for which there's quite a lot of interest at the moment. Now, let's look at crystallization of D-elementhal from the molten phase. So basically, we melt the D-elementhal and allow it to crystallize by cooling. Again, it's classic NMR. The solid state component is CP. The liquid state component is direct excitation with no proton decoupling. So here's on the left, the liquid NMR spectra as a function of time. On the right, the solid state NMR spectra as a function of time. And we can see at the beginning, we only have a signal from the liquid state spectrum, but then at a particular point in time, we lose the signal from the liquid phase, we gain a signal from the solid phase, and therefore all of the liquid phase is basically solidified at that time. But if we zoom in more detail on the solid-state NMR spectra, we can see that at the beginning, we have very, very broad lines as shown there, right? And that's characteristic of an amorphous phase, which then crystallizes subsequently at this point to give much sharper lines in the spectra. And here's represented spectra shown here, the liquid phase, the amorphous phase, and the crystalline phase shown there. So clearly, initially formation of the amorphous phase, and then the crystallization happens from the amorphous phase, as shown there. Another example where we get crystallization occurring from a frozen solution concerns glycine dihydrate. Now, the first reported hydrate phase of glycine was only published in 2017 by Tony Hu and others in this paper here, and this obtained by rapid flash freezing of the aqueous solution, followed by warming to a temperature around that value. So we replicated this procedure in the solid-state NMR spectrometer, and indeed, following the rapid quenching, you see a very broad signal corresponding to a frozen solution of glycine in water. But then as we increase temperature to around 210 Kelvin, we see a crystallization occurring. Now, the important point is this signal here is different from any of the polymorphs of pure glycine, and indeed using a range of other experiments, which are described in this paper, but I'm not going into detail here. We could show using HETCOR and other methods that that signal from the glycine dihydrate is fully compatible with the published crystal structure. And if we leave the glycine dihydrate for a longer time and also heating it, there's a transformation occurs to the beta polymorph of anhydrous glycine as shown there. So I want to conclude in the final minute or two with a couple of other areas of application. More recent work here looking at the formation of a metal organic framework by in situ NMR. And a particular example is shown here. This particular linker, when reacted with nickel nitrate, produces this NFM 500 MOF containing nickel. And although we had carried out a classic NMR experiment, I'm going to focus on the liquid state proton NMR component of that classic NMR experiment. We're going to look at the proton NMR spectra as a function of time at different temperatures with a view to gaining kinetic information on the MOF formation process. So here we see at different temperatures, as indicated, the evolution of the liquid state proton NMR spectrum. And we're going to be focusing in particular on the three different proton environments in the linker, as shown here, and how they evolve as a function of time. And integrating the signals for these different proton environments, and then looking at how these intensities vary as a function of time. These have then been fitted using what's called the Gualtieri model, a very commonly used model to describe kinetics of the formation of microporous materials, MOFs, and so on. And we see very, very good fits to the data as shown here. We can extract from those fits information such as the rate constant for nucleation, the rate constant for growth, and another parameter which gives the standard deviation in the mean nucleation time. As I said, very good quality fit as shown there, giving us detailed insight into the crystallization kinetics of the MOF material from in situ NMR data. So here we can see the nucleation probability at different temperatures from the Kn and the D parameters. So here we can see at high temperature, very sharp and narrow window where nucleation occurs. At low temperature, much broader period of time, much longer period of time over which nucleation occurs. And we can also look at activation parameters for the nucleation growth from the temperature dependence of these two different rate constants. Anyway, so what I want to conclude with in the final minute is work we've been doing in collaboration with Julia Mollica and Pierre Thureau in Marseille, where the basis of this collaboration was to think of ways in which we could exploit DNP NMR to study crystallization processes. And I want to just highlight very briefly the basis of the idea. So this is an ex situ study of the evolution of crystallization. And as I say, the objective is to benefit from the intensity enhancements from dynamic nuclear polarization. And one of the motivations here is to be able to study the very early stages of crystallization where the amount of the solid form is very low. But secondly, to be able to, to allow us to carry out more sophisticated NMR measurements, maybe two-dimensional NMR measurements, which we cannot do in the time scale of the in situ studies I described earlier. So anyway, the basis of the idea is the following. Again, we start off with the crystallization solution at high temperature. We subdivide that into several different NMR rotors, and we allow the crystallization to evolve in parallel in each of these NMR rotors. And then after a particular period of time, T1 in this case, we'll then quench that crystallization solution in the NMR spectrometer and measure the solid-state DNP NMR spectrum. We leave the next one for a slightly longer time, quench it again, and study the NMR spectrum, and so on. And the point is, after you quench the crystallization system, there's no further evolution of the crystallization. And as I said, we can then do very detailed NMR experiments at the low temperature on these quenched crystallization systems. So we get the time dependence by looking at each of these snapshots of the crystallization, each one quenched at a different point in time during the crystallization process. So very briefly, here's just one example from this paper shown here, crystallization of glycine from water. After 10 minutes in this experiment, after quenching, we can see we have predominantly the beta polymorph, some amount of the alpha polymorph, and a lot of the uncrystallized glycine in this frozen solution. As we go to a longer period of time, we can see the transformation of the beta polymorph to the alpha polymorph, and then even longer time, we actually have only the alpha polymorph and no significant amount of the uncrystallized glycine in this particular case. So this shows us how we can benefit. This is really just touching the tip of the iceberg and what was done in this study, but utilizing DNP in this area of exploring crystallization processes. And just very finally, a more recent paper as part of this collaboration was looking at crystallization of glycine in confined porous materials. And the big advantage here is that the polarizing agent can be embedded within the porous material and therefore will not interfere with the crystallization process. One of the questions, although we do various control experiments, but one of the questions here is, does the polarizing agent influence the pathway or the mechanism or the kinetics of the crystallization process or not? And that's avoided in the crystallization here. So that was really just touching upon another opportunity to study crystallization processes by NMR. So I hope I've given you an overview of some of the possibilities, some of the progress in this field for studying crystallization. And I want to end again by thanking the organizer for the opportunity to give this lecture, but especially to thank my research group and very important collaborators, as shown there, and funding organizations for allowing this work to be carried out. And thank you very much for your attention.
Since we are considerably over time, if anybody would like to ask questions of Professor Harris, please do. Otherwise, I know that we have only a short break before the next session starts. So please stay behind for any of you who would like to talk to Professor Harris about his presentation. Thank you all for an excellent session this morning.